Hello, everyone, and welcome to today's talk on GMP production of messenger RNA. I am Nathan Spangler, Director of Innovation and Strategy for Aldebaran's messenger RNA platform. Today, I'll cover manufacturing considerations for mRNA-based therapies and vaccines entering clinical stages of development. Before jumping into that, I'd like to begin with some background on Aldebaran. We are a contract manufacturing organization that specializes in the production of nucleic acid-based medicines and critical starting materials, as well as the production of enzymes for gene editing and for mRNA manufacture by in vitro transcription. Aldebaran was founded in 1998, providing plasmid DNA for research use only. Over the years, our services expanded to satisfy a wider and more demanding range of needs to include preclinical, clinical, and commercial stage manufacturing. Aldebaran is headquartered in Fargo, North Dakota, where manufacturing for clinical and commercial uses takes place today. We can provide plasmid DNA and messenger RNA in single batch sizes that currently top out between 10 and 100 grams. The photo in this slide is a close-up shot of our current manufacturing facility, B1, with 70,000 square feet completed in 2018. The next shot pans out to show our newest facility, B2, under construction behind B1 in the foreground. B2 adds 189,000 square feet to meet the rapidly growing demand for genetic medicines and vaccines, and B2 manufacturing capacity begins coming online this summer. Notably, it features a 6,000 square foot mRNA production suite designed to handle flammable solvents used in reverse-phase chromatography, as well as needs for BSL-2 containment when, for example, producing RNA designs based on alpha viruses and oncolytic viruses. Our expansion was designed in consultation with regulatory agencies, industry professionals, and most importantly, our clients. While Aldebaran has a variety of services to offer, today's talk focuses on plasmid DNA and mRNA manufacturing. Over the past 22 years, working with hundreds of clinical and commercial stage clients, our manufacturing services have supported end uses ranging from critical starting and raw materials to direct injection APIs. Currently, the majority of plasmid DNA manufacturing at Aldebaran provides raw materials and intermediates for viral vector-delivered gene therapies and gene-modified cell therapies. However, the sudden interest in mRNA-based therapies and vaccines is rapidly increasing manufacturing demand for DNA template and in vitro transcribed mRNA. mRNA is interesting for its ability to deliver transient genetic information, which instructs the body to make its own medicine or vaccine in the form of an enzyme, antibody, antigen, or other type of protein. The possible range of applications is broad, and potential is great, as we are now seeing in experimental therapies and rapid responses to emerging pandemic threats. Excited by the possibility that mRNA will rapidly fill critical gaps not met by traditional small molecule drugs, available biologic treatments, and emerging gene therapies, Aldebaran is building a comprehensive mRNA manufacturing platform that includes master cell bank and DNA template production, as well as the enzymes needed for cell-free mRNA synthesis. Production of these key materials to supply our GMP mRNA manufacturing takes full advantage of our progressive quality grades, which are a great fit for early clinical programs. Here, production of the master cell bank and DNA template often begin in Aldebaran's GMP source grade, a faster and more cost-effective option when full GMP isn't needed. GMP source works by adopting the most relevant features of GMP manufacturing, including traceability, document control, and materials segregation. GMP source is ideally suited for manufacturing ancillary and critical starting materials, and it's a great advantage to our clients transitioning from preclinical to clinical stages of development. Each mRNA manufacturing program has unique requirements for support and direction in reaching the clinic, and Aldebaran has multidisciplinary teams of experts to understand client-specific requirements and engineer manufacturing solutions that fit. Aldebaran can manufacture mRNA using our standard process, client-specific processes we develop, or a client process transferred to us. There are 4 stages to a new mRNA program, beginning with exploration or tech transfer, depending on the amount of knowledge that is available and the readiness for scale-up. In the first stage, RNA manufacturing knowledge is gained to enable the scale-up process development in stage 2, where the master cell bank and template DNA are also produced in GMP source or GMP. Stage 3 begins with the full-scale RNA engineering run in GMP facilities and concludes with the finalization and approval of specifications and manufacturing documentation for GMP manufacturing, which happens in the fourth and last stage, ending with the delivery of mRNA drug substance, manufacturing batch records, and a certificate of analysis. A typical RNA manufacturing program spanning these 4 stages can run 9 months or longer, but this timeline can be compressed for urgent needs. Because messenger and self-amplifying RNA can be synthesized using enzymes rather than cell culture, the manufacturing process is simpler, more scalable, and more easily expedited than for other biological medicines. This has been a key advantage for rapid responses to emerging threats and unmet medical needs. In response to COVID-19, for example, we're helping clients begin clinical testing in a fraction of the time needed for conventional vaccine technologies. We have seen that the ability to rapidly scale up vaccine manufacture can be limited by plasmid DNA template and enzyme supplies, and our ability to produce these has been an important advantage to our clients. Most new mRNA manufacturing programs at Aldebaran begin with plasmid template generation. Although we do not design templates for our client, we can offer general guidance to improve manufacturability for the intended use. 3 important elements to mention are the template region, the origin of replication, and the antibiotic selection. The template region is the most complex of these to figure out, and there are multiple things to consider. It should include elements needed for mRNA synthesis by in vitro transcription and for optimal mRNA expression and stability in vivo. In the context of plasmid manufacturability, templates encoding poly-A tails require extra care because tail retention during plasmid manufacturing is a concern, and we recommend that the tail length not exceed 100 bases for better plasmid stability. Poly-A tail retention is confirmed by sequencing, and during cell bank production, host strains and growth conditions are screened for optimal stability. The linearization enzyme on the 3' end of the transcribed region is another feature to think about carefully. Ideally, the restriction enzyme chosen for linearization will be economical, easily sourced, and available in an animal-free formulation. It is also recommended to choose an enzyme that cuts, leaving a 5' overhang or blunt end for runoff transcription. The origin of replication is important for plasmid manufacturability, and a high copy number can make purification and scale-up easier. In response to regulatory concerns over residual beta-lactams, kanamycin resistance is preferred, though we are able to manufacture plasmid templates with ampicillin resistance. To avoid antibiotic selection completely, companies such as Nature Technology have enabling plasmid technologies available. Back to the template region, but this time to focus on the elements important for in vitro transcription and in vivo translation. For mRNA synthesis at Aldebaran, the template must have a T7 or sp6 RNA polymerase promoter to drive in vitro transcription. The 5' untranslated region between the T7 promoter and coding sequence influences translational activity in vivo, and the Kozak sequence is usually part of this design element. On the other side, the 3' UTR and poly-A tail play important roles in the regulation and stability of the synthetic RNA in vivo. For example, instability elements could be encoded that accelerate mRNA degradation. Conversely, there are 3' UTR sequences known to enhance stability as the poly-A tail does. In the template designs we usually see, poly-A is most often the only 3' UTR feature. For more on UTR designs for therapeutic use, there are several good publications which I'd be glad to recommend. The template coding sequence will encode a single protein for expression, or a more complex design for a self-amplifying mRNA, which is more challenging to produce due to its greater length. Finally, there must be a site for plasmid template linearization as discussed. This site does not need to be unique as long as the secondary site is not within the template region or in the backbone near to the primary cut site or the T7 promoter. With mRNA-based therapies and vaccines, there's been an upward trend of heightened focus on CMC, which impacts release testing and specifications for the DNA template as a critical starting material, and the mRNA drug substance for a range of uses, from ex vivo cell therapy to infectious disease vaccine. Per the most recent FDA guidance on CMC, it's recommended plasmids be attested to ensure identity by sequence verification, as well as by restriction digestion. For the DNA template, agencies have noted the importance to understand the ability of the process to minimize residuals like endotoxin, host genomic DNA, and processed proteins. This will help in establishing comparability for any modifications to the manufacturing process in later stages. We see a variety of requirements in the realm of safety assays, most notably when assessing sterility testing. Determining which method to employ is important in planning, as it will impact materials needed and the overall release timeline. There are a few options to weigh. You have traditional USP71 sterility, which will have the longest turnaround time, as well as rapid sterility or bioburden. It's really up to the sponsor to identify which is the appropriate choice for their use. Note that the test specifications highlighted yellow in this slide tend to be the ones that will impact manufacturing process and cost the most. For mRNA QC testing, release specifications can be more challenging because there is less clinical experience with this molecule type. Purity in terms of percent capping and double-stranded RNA impurity are notable concerns, and we generally look at these as characterization tests until there is sufficient justification to set a release spec. We offer a comprehensive selection of QC tests that range from compendial methods for critical contaminants to product-specific tests for identity, integrity, and purity that comply with regulatory requirements for product release. For mRNA characterization and release testing, we have a range of capabilities, either internally or through qualified partners, to run the tests needed for quality assurance. As with the Plasmid template, certain specifications, highlighted yellow here, will have an impact on manufacturing process and cost. So these, in addition to use requirements, are going to be important in defining the manufacturing process and the need for materials such as base-modified NTPs and polishing steps such as reverse phase chromatography, which will add to the overall cost and complexity of manufacturing. And I want to stress again, the choice of enzyme used to linearize the plasmid DNA template will have a major impact on cost and manufacturability, so please choose carefully when designing your template. Aldebaran's RNA production is flexible and offers manufacturing options to satisfy client preferences for capping and tailing methods and configurations. Our standard purification process involves ion exchange chromatography and tangential flow filtration, and the benefits are high downstream process recovery and scalability, and high product quality. To satisfy more stringent purity requirements for some uses, we can incorporate known purification methods such as affinity chromatography and reverse phase chromatography. We also offer custom process development, and we can scale up client manufacturing processes. Currently, our clinical grade production scales range from 1/10 of a liter to 10 liters for the IVT reaction, and we can support larger scales in our new facility coming online this summer. Enhanced capabilities will include the accommodation of downstream processes with large volumes of flammable solvents, as well as requirements for BSL-2 containment, as mentioned earlier. A major advantage to partnering with Aldebaran for your messenger RNA production is that we also manufacture the master cell bank and linear template, as discussed, and we can provide, if desired, all enzymes needed for in vitro transcription, post-transcriptional capping, and post-transcriptional polyadenylation. Our stocked enzyme products are manufactured animal component free and research grade, and we can make to order in GMP source and GMP quality grades. Research in GMP source manufacturing is supported by multiple fermenters with stainless steel trains up to 1,000 liter working volume and multiple single-use fermenters up to 300 liters that are dedicated to GMP source and GMP manufacturing. With the first messenger RNA-based therapies and vaccines now in the clinic improving their safety and effectiveness, we expect to see usage rapidly expand to include a wider and more comprehensive range of indications. Leading the way will be both therapeutic and infectious disease vaccines based on mRNA technologies. Aldebaran is preparing for the future by building comprehensive platforms and greater capacities to support RNA-based therapies and vaccines from preclinical development to commercial manufacture. We have the world's largest GMP facility for plasmid DNA and messenger RNA, and plans are underway to increase capacity by more than tenfold over the next 5 years. As a manufacturer, Aldebaran stands apart through its offering of all critical biological materials needed for messenger and self-amplifying RNA production. For 21 years, our mission has been to help improve lives through innovative partnerships and scientific excellence. Our process development and manufacturing capabilities support the innovative therapies and vaccines that will transform medicine. The biopharmaceutical industry is growing exponentially, and we are prepared for and excited about the future. Thank you for your time and attention today, and please feel free to contact me with questions.